There are several methods to measure protein kinetics. Most of them are based on measuring the degree of hydrolysis, which is defined as the amount of hydrolyzed peptide bonds. Methods to measure the degree of hydrolysis assume that a free amino group and a free carboxyl group are being released every time a peptide bond is hydrolyzed. Common to all these methods is that they quantify the increase in such released groups, and then they use a quantification as a variable to evaluate how many peptide bonds that has been hydrolyzed. The difference between all these methods really lies in how they quantify the increase in such concentrations. We have decided to use the pH-stat method in collaboration with Aarhus University, as this is a real kinetic technique, allowing you to measure the progress of protein hydrolysis instead of only focusing on the extent of protein hydrolysis. It's a well-established method, and we use the method as essentially described and published by scientists from Wageningen University. The method is well described in literature, and it has been used to measure protein kinetics for different types of raw materials and different types of products. It's a simple and reliable method, and the results found by the pH-stat method correlate well with other in-vitro methods. As you can hear by the name, the pH-stat method, it has something to do with the pH value of a sample solution. This method utilizes that an acid is being released every time a peptide bond is hydrolyzed, and that auto-titration automatically will add the required amount of an alkaline solution to keep the pH value at 8 throughout the incubation period. If we take it step by step, the first thing you do is that you mix your sample with water, and then you force the pH to 8 for all the samples that you're going to analyze. To start the reaction, you add enzymes. In our case, we add a mixture of 3 different enzymes to mimic the enzymes present in the intestine of the animal. Then the enzymes will start to hydrolyze peptides, and for every peptide bond that is being hydrolyzed, an acid will be released, which will force the pH to drop. When the pH value drops, the pH-stat automatically will add the required amount of an alkaline solution, in our case it's sodium hydroxide, to increase the pH value again and make sure that we are at a stable level of 8 throughout the 2 hours of incubation. The volume of sodium hydroxide added is being recorded at all time points, and this volume is being converted into degree of hydrolysis, and it gives you a curve representing the progress of protein hydrolysis. A huge advantage of this method is that it provides you with a rate constant, and it means that the method is not only able to tell you how many of the peptide bonds that has been hydrolyzed, it also tells you how fast they have been hydrolyzed. The faster the protein hydrolysis, the more of the alkaline solution has been added in the beginning of the reaction period. We run the pH-stat method at pH 8 because this is the optimum for the pancreatic enzymes used in the protein hydrolysis. If we lower the pH value to represent areas closer to the stomach, this will affect the enzymes because they will start to denature. As we cannot ensure consistent denaturing of proteins and enzymes when we take them outside their optimum range, this will affect the stability and general control of experimental conditions. Therefore, to ensure consistent and reproducible results, we run the pH-stat at 8, which is also commonly described and used in literature.